separates the thoracic from the abdominal cavities. And the diaphragm is usually drawn as if it's flat, but at rest it's actually dome shaped. And when it contracts, it flattens out. And then it goes back to a dome shape. So this is a resting diaphragm at a dome shaped position. Okay, so these would be the lungs. And the right lung has 3 lobes. And I remember that because the word 3 has an R in it for the right side. And the left lung has 2 lobes. Now, what we're going to look at is we're going to cut all this away and then look at a unit of a lung lobe, which is called a lobule. And that's what we're looking at over there on the model on the right, the lung lobule. There's going to be a structural unit of the lobes of the lung. Of course, you can see here that you got 1, 2, 3 lobes on this side. And I don't see where that, oh, here's the cut. So this would be 1 and the second lobe on the left side. So let's go to the lung lobule. So it turns out the lobes of the lung are structurally divided into what are called lobules. Okay, before I go over there, let's just make sure we got this straight. So the larynx leads to 1 tube, almost an inch in diameter in a human being. It's a really big tube, and that would be the trachea. And the blue stripes represent plates of cartilage in the trachea and the larger bronchi. And these plates of cartilage make the wall rigid so it doesn't collapse. It's never in your best interest for the air tubes that deliver air into your lungs to collapse. And they don't. They're held open by these cartilaginous plates. So the trachea is right there where my finger is. That's where the trachea ends. And then the trachea, where it ends, it branches into 2 large bronchi. And those bronchi then feed into the lungs and then continue to branch repeatedly. Okay, so a bronchus is a large air tube with walls that are reinforced with cartilaginous plates. And the cartilage is shown in blue. The bronchi branch into smaller and smaller bronchi repeatedly. And eventually the bronchi, they branch into tiny little tubes called bronchioles. And bronchioles don't have plates of cartilage in their walls. So on the model, when we get down to tiny tubes without plates of cartilage, those would be the bronchioles. And they're less than 1 millimeter in diameter. So a millimeter would be the smallest gradation on a metric ruler. So we're talking really tiny tubes, just to give you a sense of the scale here, because these tiny tubes here would be bronchioles. Okay, this one right here with the blue colored plates of cartilage is a small bronchus. And then it's going to branch into these bronchioles. And the bronchioles go into what's called an alveolar sac. So let me show you these alveolar sacs. This is an alveolar sac. Okay, here's another alveolar sac. There's an alveolar sac. That's an alveolar sac. That's and maybe this is one alveolar sac. But this cluster of little air sacs, all of it taken together. And there may be a couple hundred little air sacs associated with an alveolar sac, make up an alveolar sac. Okay. You'll see salmon-colored stripes, sort of a brownish-red stripe, around the bronchioles. Those are smooth muscle cells. And when they contract, that's an acetylcholine parasympathetic effect, and it makes the tube smaller. And when they relax, they release their grip on the tube. The tube gets bigger. That's a sympathetic effect that would kick in when you exercise. If I injected you with epinephrine, you would bronchodilate as those smooth muscle cells relax. And these tubes get bigger, and that means that more air can go into and out of the little alveoli or air sacs of the lung. One of these bubbles is an alveolus. An alveolus is just a fancy name for an air sac. And when you change the US to an I, it becomes plural. That would be alveoli. So this is a cluster of air sacs, or a cluster of alveoli. Now, every single one of the air sacs, every one, is covered with a set of capillaries called a pulmonary capillary network. Now, I think last week, didn't we go over this? And I mentioned that just for the sake of, keeping the costs reasonably low, because these are all hand painted. You don't have the pulmonary capillaries over these alveoli, but they would be there in a real individual. Okay, the coloring scheme. We're used to arteries being red and veins being blue, and that is true in the systemic circuit, but everything is reversed in the pulmonary circuit. So the blood vessels that we see here, most of them are going to be associated with the pulmonary circuit. So you have a blue-colored artery and a red-colored pulmonary vein. Pulmonary artery, pulmonary vein. And these branch, as they branch into smaller ones, into pulmonary arterioles. Of course, these red ones here would be pulmonary venules, pulmonary vein, pulmonary artery, pulmonary arterioles, feeding or draining the pulmonary capillaries of the alveoli. So this model does contain some systemic blood vessels, and you can see them down here, where you'd have a red arteriole feeding a capillary bed that's drained by a blue-colored systemic venule. So these are systemic blood vessels. And the coloring scheme is a little bit different. So if you notice that, see the red right here, that color red right there? This is actually a systemic blood vessel that's feeding these blood vessels down below. So they've tried to have a different shade of red. So this is a really dark, maroon red for the pulmonary, and sort of a lighter one, maybe with a little more orange in it or something like that for the systemic. Okay. So wherever you see the little red fibers, red stripes, those would be smooth muscle cells. Okay. There's no question this is bronchus because it has plates of cartilage. And there's really no question when you get down to there or here that you don't have the cartilage. That's the key. And those would be bronchioles. And when I test you on it, I'll try to make sure I mention something like that. All right. Over here would be alveoli. So the bronchioles, for the most part, if they're less than 1 millimeter in diameter, we're talking microscopic. So this is something that you wouldn't be able to see with the naked eye, because there's something like 150 million air sacs per lung, just to give you a sense of scale. We're really down at something that's very, very small right here. And this clearly would be microscopic. In fact, capillaries are so small, these capillaries, that they were discovered long after people knew about arteries and veins. It just took a long time to get a microscope with enough resolving power to be able to see them. But this would be the air sac, and the wall of the air sac is made up of simple squamous epithelium, and there's 2 types of epithelial cells. Type 1, which makes up most of them, and type 2 that you can see with a little brown spot, and those make a chemical called surfactant, and I'll tell you about those in class. Okay, the alveoli are covered with capillaries, pulmonary capillaries. Okay, remember the coloring scheme is reversed. So if we've got red, it's got to be a pulmonary venule. If we got blue, it's got to be a pulmonary arterial, right? Okay, so let's scrape away the simple squamous of the alveolar wall so that we can see the capillaries on top of it. So we just scraped away this stuff, and now we're just looking at the capillaries. And where we pick up blue, we know that's got to be an arterial. And the red tells us systemic, pulmonary caps, it's got to be a venule, okay? So just don't get confused. And the black would be elastic fibers that lace over the alveoli throughout the lung, and that makes the lungs really stretchy. So it turns out that when you inhale, it's like blowing air into a balloon. So if I want to fill a balloon up with air, I have to blow up the balloon, and the balloon expands. But if I don't tie that balloon off and I just let it go, then the stretched elastic rubber molecules in the wall of the balloon, they will recoil back to their original length and blow all the air out of the balloon. Does that make sense? Because we're going to find that it takes energy through the contraction of skeletal muscle to suck air into the lungs, but as they expand, they gain potential energy with the stretching of those elastic fibers, and the air in the lungs will blow out of the lungs when you exhale just by what's called elastic recoil. Okay, so do make sure you take a look at this model. All right, let me show you something on page 552, and we can do this really quick.